Hello, everyone. Welcome to my Spring 2020 Academic Partners Project presentation, The Chemical Analysis of Lavender Oil, Natural versus Synthetic. I did this project with Dr. Rodney Austin and Dr. John Stahl. I originally didn't plan to be doing this presentation virtually, but I'm still really excited to show you guys what I did this semester. So I just wanted to start off with a little intro. My name's Felicity Orndoff, and I'm currently a junior at Geneva College. I'll be graduating next spring in 2021 with a dual degree in chemical engineering and chemistry. In the picture on the top left is me and some of my coworkers from Sonneborn. I worked there last summer as a quality control lab intern, and it was a great experience. I really learned a lot. This summer, I plan to go down to Houston, Texas to work in the nutrients lab at Konica North America. So I'm really excited for that. I also play on the tennis team here at Geneva. And I also like to go on adventures. And you can see in the bottom left picture is me and my family going scuba diving in Florida over spring break. I actually scuba dived for like 2 minutes and a stingray almost touched me and I screamed and ran out of the water. And the picture on the top right is of me and a camel in the city of Jericho when I went on a trip to Israel and after that Egypt this past Christmas break. And I also have been playing the ukulele for a few years now and I'm learning the guitar. So before I begin, I just wanted to address the natural product controversy that seems to be taking over commercial life. And as you saw the title to my project, I'm sure this popped in your head of the last time you were at the store and you picked up a label and read that something was all natural and vegan and healthy, and there's just no proof. How do you know that that natural shampoo is better than a synthesized shampoo, even though it's $5 more for some reason? That should make it better. I just wanted to talk about the two extremes to this spectrum that we seem to find ourselves in between and just present a healthy case so we can move on with my research. So, starting off, let's talk about the natural product maximalists. They tend to have a complete preference of natural products over synthetic. They'll be the natural product purchasers. They're the ones to buy that $5 bottle of shampoo over the $1 bottle. And typically, they have a blind acceptance that it really works because there's been minimal research to back most of these products. They typically reject synthetic drugs, physician advice, and even vaccination in extreme cases. On the opposite side of the spectrum, we have the minimalists, or the skeptics, I like to call them. They think that natural products will never replace synthetic. They're skeptic shoppers because they seek this evidence that, you can't trust these companies because it could just be an advertising ploy. They also tend to reject natural remedies, unprofessional medical advice, and essential oils. So I think a good place to stand in this controversy is somewhere in the middle, just so we can have an unbiased research on natural products. By having equal support of natural and synthetic compounds and recognizing medical advice and previous chemical research done, we can really find the truth behind which is better, natural or synthetic. Okay, now that our biases are completely gone, we can start into the background of chemistry and nature. So we're going to start by being very general and work our way into being really specific. So I just wanted to tell you guys about the terpene compound as shown in that little circle to the left. So these guys are really common. They comprise approximately a third of all naturally occurring chemical compounds. So they are everywhere, and they're super complex because more than half of substrate carbon atoms undergo changes in bonding, hybridization, and stereochemistry. This makes them very hard to synthesize, especially to get the correct stereochemistry, which is the orientation of the molecule. And they can undergo cyclization, which is typically multi-step, and this reaction occurs in the roots and branches of a plant, and the final product we desire is in the leaves. So you're probably thinking, Felicity, you're not a biochemist. How could you possibly know? Like, what's so special about this terpene compound? Well, let me show you. So the alpha terpenyl cation at the bottom of the molecule allows it to cyclize into hundreds of different compounds found naturally in plants. And this is just a handful, but I want to focus in on this plus bornyl diphosphate in the bottom left. So if you look at the structure of bornyl diphosphate, you can see that it cyclizes to form a sort of carbon bridge directly in half of the molecule. And this interesting quaternary structure matches a lot of other terpene compounds, such as isoprene. This proves that there's a universal dimerization mode for this alpha beta architecture. It provides more stability compared to compounds such as geranyl diphosphate in the top of the diagram. And these compounds are not necessarily final products. Usually, the alpha terpenyl cation will form one of the many compounds, and it may cyclize into one of the other compounds across the table. So, a great example reaction is the natural biosynthesis of camphor in plants. And it starts off with that geranyl diphosphate compound as seen on the last slide, and it fully cyclizes to form the plus bornyl diphosphate with the quaternary structure. It then substitutes the diphosphate group for an alcohol, and that alcohol can be oxidized to form camphor. So, due to the high demand of these natural products, several synthesized alternatives have been developed. This one specifically involves a few more steps than the natural synthesis, but overall is simpler because it starts with alpha pinene, which is an already cyclized molecule. It then forms camphene and isobornyl acetate, and then we have our similar substitution of the alcohol group and oxidation to plus camphor. But notice now we have 1R plus camphor, which is stereospecific, and we're unsure if this truly matches the natural biosynthesis of camphor. So this is where I'm going to focus in on my research. So you probably noticed that both syntheses concluded with some sort of conversion between camphor and borneol compounds. And these two differ only by that reduction or oxidation to create two very different molecules. Camphor is actually toxic and it's lethal over a dose of 3.5 grams, causing congestion of the gastrointestinal tract, kidney, and brain. A plant would usually create camphor to defend itself and protect it from animals taking its borneol supply, which is non-toxic and beneficial to its health. And it has been clinically proven even to have anti-inflammatory and antibiotic qualities. From this, we can conclude that a higher borneol content actually improves the quality of a natural product we're trying to create. And specifically, I want to look at lavender oil because camphor, borneol, and a third compound called linalool are the three most abundant, so we can better see this conversion of camphor to borneol. Most of you are probably familiar with lavender oil because it's one of the most popular natural compounds even purchased by most minimalists. And that's due to the high content of linalool. This compound is not cyclized. In fact, it's a straight chain consisting of an alcohol group and a few double bonds. This compound causes natural drowsiness as an anesthetic of sorts, and it has also been clinically proven to be antimicrobial and antiparasitic. So, from this, we can conclude that the entire lavender compound has been proven antimicrobial as to previous research. So, that was a lot of organic chemistry just to show you how I decided to do this research on lavender oil, because it truly is a powerful perennial with hundreds of active monoterpene compounds, 60% of those being camphor, borneol, and linalool. And the craziest part is, there's been very limited research done comparing these two syntheses of the natural and the synthetic. And this research isn't necessarily that difficult because all we have to do is quantitatively analyze camphor, borneol, and linalool content, and from that we can infer the quality of the oil. Did I say necessarily not that difficult? Well, it was kind of difficult, but let me explain to you my plan through laboratory analysis. So I like to call this my master plan, consisting of two phases, extraction and instrumental analysis. Unfortunately, I was only able to complete phase one before our semester got moved online, but I'm still going to lay out my plans for phase two and for the future. To begin phase one, I needed to buy some lavender, and that can be a little difficult in Pennsylvania in the middle of winter. But after a little research, I found Lavender Green Farm in Knox, PA, which was actually owned by Geneva alumni, which was pretty cool. And from them, I bought a dried organic lavender sample that I could run through a steam distillation. Through this distillation, I could isolate the oil as part of my natural sample that could be compared to standards of camphor, borneol, and linalool in phase two. This includes using both gas chromatography and mass spectrometry to analyze for both qualitative and quantitative measures of the oil sample. And hopefully, down the road, I can do some sort of biochemical analysis to analyze for antimicrobial qualities. So now I'd just like to elaborate on what exactly each of our three instrumental analyses can do. So first, for identification purposes, we would use mass spectrometry, and this will allow us to have a qualitative analysis of the oil to break it down into each of its components by mass. This highly pressurized system creates a suction that can pull the vapor on top of the oil liquid into a quadrupole mass analyzer, where heavy atoms will drift towards the negative rods and the more light atoms shift towards the positive rods, creating a change in voltage that can be picked up by the detector. Then, by the use of gas chromatography, we can first run the standards of linalool, camphor, and borneol, and then run the naturally extracted sample and compare the peaks for quantitative analysis. Typically, for these types of oil compounds, you would use a polar column, such as the Supercoax 10, as shown in the picture there, and the following is the order of retention time in minutes, so it's a fairly quick chromatography, with camphor being retained for 25 minutes, linalool for 27, and borneol for 35. And finally, by the use of agar plates, we can determine the antimicrobial qualities of the oil. And this is done by creating a dilution of oil with ethanol, placing it on a plate in a petri dish with bacteria, and measuring the diameter of the inhibitory zone created by the oil. And by that diameter, we can determine how effective the oil is at killing the bacteria. So I chose to extract my natural sample through steam distillation because it provides isolation without destruction of these delicate compounds. It has a relatively simple setup and a low cost compared to some other methods. One I found was supercritical fluid extraction that is typically done in only high-end research laboratories. And this method still provides the isolation of my desired compounds, but with a few limitations. Some of those being the hydrolysis of water-sensitive materials, possible degradation of thermolabile compounds, the incomplete collection of molecules with pharmaceutical properties, or the inability to collect compounds with low vapor pressures. It should be noted that all three compounds I'm looking at have very high vapor pressures, so this shouldn't be an issue. I also plan in my future work, possibly as a senior design project, to enhance this steam distillation process because it tends to have a very low percent yield of oil per distillation. So, with the assistance of Dr. Austin and Dr. Stahl, I was able to create this steam distillation apparatus. And I'm not sure if the picture does it justice, but this apparatus stood probably close to 3 feet high off of the lab bench. Well, clamped, of course. And after building it, I was so nervous to try a distillation. So I did a preliminary distillation on orange peels to try to extract orange oil. So one of the most important pieces of the distillation apparatus was the Soxhlet Receiver. This receiver collects the oil and hydrosol. So a little vocabulary. The oil is the high purity lavender oil that comprises the top layer of the distillate, and the remaining hydrosol is the heavy bottom layer that contains all of the water used to distill the biomass, and this layer contains water-soluble compounds left over from the oil. This receiver provides an atmospheric vent, as no vent can be placed before the receiver because most of the distillate could escape. So we still have to provide some sort of atmospheric vent so the pressure doesn't overload on the system. So by this Soxhlet, you can see the skinny tube running from the top of the receiver out to the bottom in the beaker. We're able to vent the system without losing any product. The only issue is the somewhat large width of the Soxhlet, and that makes the oil interface kind of difficult to make out. Thankfully, all of my nerves about extraction were relieved because I was able to successfully extract orange oil. However, I did learn a lot from this preliminary distillation, and I made the following modifications before I distilled my lavender. The first involved changing the wire gauze stopper in the biomass flask to glass wool, and I did this to provide a cleaner steam transfer to the biomass, as well as an easier cleanup system for afterwards. To improve the oil film visibility that we talked about from the Soxhlet receiver, I added a portion to the apparatus, including a separatory funnel and a ring stand, so I could drain out the distillate from the Soxhlet and carefully funnel it into the separatory funnel. In this funnel, it was really easy to see the oil to water interface. Ta-da! This is my final steam distillation apparatus I use to extract my lavender oil. So it starts off on the left with a hot pad that heats up the boiling flask containing a large amount of distilled water. When that boils, it generates a steam that rises through the glass wool stopper, and the glass wool transfers the steam to the biomass flask, where that heats up the dried lavender buds, and they release the vaporized oil compounds along with the steam as it travels through the still head and finally condenses and creates a distillate drip in the Soxhlet receiver. From here, that distillate can be drained off and poured into the separatory funnel, where we can better distinguish that oil interface. Yeah, all that chemistry talk. You guys really care about the results, right? Well, I was successful in extracting lavender oil, and let me show you more. So here's a great before and after distillation picture of my biomass, and it's very obvious that these lavender buds lost a lot. They lost their color, their odor. Um, they probably wouldn't even be recognized as lavender anymore afterwards. And then on the bottom, you can see my two layers, the oil phase and the hydrosol phase. And I was able to drain off a lot of hydrosol, but unfortunately, I couldn't drain it all off without risking draining some of the oil. So here's some of the data and observations from the steam distillation. I ran each dry mass in 2 batches for a total of 20 grams of lavender. The first batch I distilled for 1 hour 17 minutes. The second batch was about 1 hour and 2 minutes, for a total distillation time of 2 hours and 19 minutes. The initial dry lavender had a light purple color and a very strong lavender aroma. The wet lavender afterwards had a light brown color and little to no odor. The lavender hydrosol I collected had a mild floral and kind of a sweet aroma, and it was very cloudy because there were a lot of dissolved compounds in the water. The final lavender oil extracted had a sort of yellow tint, and it was extremely volatile. After I did my distillation, I smelled like lavender for days. It had this pungent floral odor, very similar to the dry lavender, but way more intense. And unfortunately, I couldn't do the percent yield data because I had not successfully isolated the oil from all of the hydrosol before the project got cut short. This percent yield calculation would have consisted of a pure oil to dry biomass ratio. And it's a lot easier said than done to separate these two compounds because the oil is so volatile and there was such a small amount on top of the hydrosol. So some of the drying methods I considered was using magnesium sulfate, a 3 angstrom molecular sieve, or possibly a rotary evaporator to isolate my oil. So the last laboratory analysis I was able to attempt was mass spectrometry. And I was able to do this on my wet oil because the capillary only analyzes for vapor on top of the compound. So my extremely volatile oil should have been detected by the capillary. However, I was having some issues with this as the capillary could have been clogged with dust or could not have detected the very small amount of vapor created by the oil. So I then tried one of my standards. I believe I chose the linalool standard. However, I don't think the sample was prepped well enough as not enough vapor was generated in my beaker before it was analyzed. So I only had one mass spectrometry attempt. I would have loved to have gotten more, but I was cut off in between. So at this time, I'd like to make some partial conclusions, solely based on my steam distillation technique. So the primary flaw was the low oil yield that could be solved by increasing the biomass or decreasing losses, possibly due to heat loss. By providing some sort of insulation around the flask, we might be able to generate more steam through the biomass. And this process was also a very lengthy extraction. Most oil is extracted in the first 30 minutes, but to assure all of the oil is extracted, we want to run for 1 or more hours. So this can be some lengthy laboratory time. But one of the more positive effects of the method was the purity, and this can be determined by the very unique, strong aroma of the final product. And I was able to smell both my naturally extracted oil and compare it to a purchased synthetic oil and a purchased natural oil. And the extracted version was so much stronger. And this hints to possible dilutions even in the natural commercial oils. So, even though I wasn't actually able to perform any antimicrobial studies, I just wanted to share with you guys one that I found. So, this study was done to determine if essential oils could take the place of some chemical food preservatives. And they analyzed 15 oils, unfortunately, none of which were lavender. However, they did analyze our linalool standard, and most of the oils that were tested did include a high content of linalool. So, by diluting each oil with ethanol and placing this solution on a paper disc, they were able to look at the inhibition diameters on agar plates. So, by measuring these inhibition diameters of each oil, we can determine how effective each one is at killing bacteria. And as seen in the picture, not every oil had the same inhibition diameter. However, each showed a small diameter, suggesting that there are principal bioactive constituents in essential oils. So, therefore, they may serve as effective natural alternatives in food preservatives. This study can easily be done on lavender oil, and we can also use different bacteria to determine other antimicrobial or antibiotic effects depending on what ailment we're looking to cure. So this is the saddest slide I'm going to show you because unfortunately I could not perform any other instrumental analyses. And I don't really have an answer for you of which one's better, natural versus synthetic. But this is kind of a to be continued as my senior design project, so I'm really determined to get you guys an answer. And even if I don't find one next year, I want to continue to look into this, and I found new passions in natural pharmaceutical research to maybe create a simplified extraction process and hopefully find a greater purpose in this natural product chemistry. So this concludes my presentation, and here are the references that I used. And I really hope you have a question because I am more than prepared to answer them. So please, after you've finished watching this, feel free to call me, text me, or email me any questions. And if your curiosity is as crazy as mine, you'll like Romans 8:19, for the creation waits in eager expectation for the children of God to be revealed. Thank you so much for watching this. Goodbye.